Good evening, Ishika. How are you doing? Has the school completed the genetics chapter? Have they begun any new chapter or still stuck with the genetics? Okay. So we'll finish it today. So last time we had talked about the chromosomal mutations, right? This time we'll be talking about. Okay, good. So you can send any worksheets to me if you want, Ishika. Like so that I can make you prepare even better in a better way. If you have, you can send your worksheets to me. If you want any kind of assistance, you can definitely ask. Send those questions on my number only directly. I'll help you out. Mhm. So we'll be talking about the Mendelian disorders today. So okay, okay. Whenever you have, if you need any kind of help in any topic also, like you can contact me anytime. Any conceptual understanding. Yeah, sure, sure. So any conceptual understanding, if you need any help or any questions or worksheet, anything you can send. Okay. So now we'll be talking about the Mendelian disorders here. So Mendelian disorders mean we will not be tampering with chromosome number. Okay, chromosome number would remain constant. Okay, so here we are not tampering with chromosome number. No manipulation of chromosome number. The chromosome number stays okay, constant. Okay, what happens is they have mutation. Okay, and one more thing, if I'll tell you, like these things are used interchangeably. Sometimes they say mismatch, sometimes they say mutation. Both the words are not equivalent. Okay, there is difference between, I'll tell you, mutation versus mismatch versus another word comes up in biology that is known as SNP, single nucleotide polymorphism. So these three terms have distinct differences. They should not be used interchangeably, but most biologists they use it interchangeably. There is one mismatch, mutation, SNP. They look similar. Okay, the explanation sounds similar, but there is a distinct difference. So the difference is just take an arbitrary genetic sequence from 5 prime end to 3 prime end, from here 3 prime end to 5 prime end. Okay. So this is an arbitrary DNA sequence. Now suppose what happens is the DNA polymerase enzyme, which will go for the duplication of DNA or replication of DNA in the space of the cell cycle in case of eukaryotes, okay, or the prokaryotes that do not have the cell cycle event. So DNA polymerase there also will go for the template strand. So both these strands are taken as template. Okay, suppose 3 to 5 prime end is taken as a template, and now a new daughter strand is produced from 5 prime to 3 prime. Okay, this is a template now. Suppose what happens is the polymerase, the DNA polymerase enzyme, okay, which makes the new or which synthesizes the new strand by adding up the DNTPs, means DATP or DTTP or DCTP or DGTP. Okay, so the reaction will be, it will become D, means deoxy, D means deoxy, GMP plus PPI plus energy. This will lead to the phosphodiester bond formation. This is specific energy, right? Now, these are all nucleotide. Nucleotide means the base, the phosphate, and the sugar, right? So suppose this is becoming its complementary. So A, T, G, C, C, okay, then T, A. Instead of G, it became A here. Now it became the same T, A, T, G. So this is known as a mismatch now. Okay, only if one of the DNA strands has got a different nucleotide or a different base. Okay, nucleotide means base. The sugar and the phosphate is still the same, different base, which does not match with the template. The other one, when it was coded, okay, the other one 3 to 5 prime one is the same. Okay, suppose the other 3 to 5 prime one when it is coded, 3 to 5 prime one, it is the same. It is T, A, C, G, G, A, T, C, A, T, okay, A, then C. This one is the same. This one, the new daughter strand, okay. This daughter strand is the same. This daughter strand has got one mismatch. That is why it is known as mismatch. It should have been G, right? But it is A due to the fidelity problem, due to the, you can say, intrinsic, you can say, drawback in the in the DNA polymerase enzyme, which is then taken care of by the exonuclease activity that will come later on in your course, and the DNA repair enzymes are also there. But this is known as a mismatch. Okay, this is known as a mismatch. But if this happens here also in the second strand also, in both the DNA strands, if this happens, then it is called a mutation. If a single strand contains this specific defect or change in base sequence, it's a mismatch. If both the strands contain it, if both the strands comprise of that specific, you can say, mismatch, two mismatches will make a mutation, one mismatch will make a mismatch only. That is mutation and mismatch, the difference. Okay. So mutation will be like this. So both these strands, suppose it is A, T, G, C, C, G. Here, T, A. So suppose this is a mutation. Suppose here it should have been A, here should have been T. Now first this became G, now the complementary became C. Now this one is a mutation, not a mismatch. Both the DNA strands have got a different base instead of what it should have been. So this is a mutation. Okay, only one of the bases, if they have, or it had been changed, altered, then it would have been a mismatch. And now when we come to a population, okay, that comprises of the individuals belonging to the same species, so in a population, if this mutation is present, it is prevalent in more than 1% of the population, then it becomes single nucleotide polymorphism. If it is prevalent in less than 1% of the population, if this same mutation, it is known as mutation. So mutation and single nucleotide polymorphism are molecularly same, but only the prevalence in the population, the percent prevalence in the population decides what it will be referred to as, what it will be called as. Whether it will be called as a mutation or an SNP. So the acronym is SNP for this one. Okay. So this is the main difference between a mismatch and a mutation and an SNP. Okay. So I hope you have understood this one. Now we can go for Mendelian disorders. So we know that we have 23 pairs of chromosomes. Okay, 23 pairs of chromosomes. Now, in this 23 pair of chromosomes, 23 pairs of chromosomes, the first 22 pairs are known as autosomes in humans. We talk about Homo sapiens. The last pair, it could be XX or XY in general cases could have been XXY, triple XY, triple X syndrome that we had talked about, X naught, Turner's XXY, Klinefelter, all those things can happen. But here we are talking about only Mendelian, means the chromosome number will stay constant. So either XX or XY, this one pair means two chromosomes are known as allosomes slash sex chromosomes. In many books they will refer to as sex chromosomes, but the technical term is allosomes for this one. Okay. Now considering Mendelian disorder, you have to keep in mind a disease could be either autosomal or allosomal. First of all, this one. A disease could be either autosomal, means it has affected the autosomal chromosome, the mutation, or allosomal. Okay. Under allosomal, it could be X-linked, means mutation happening in the X chromosome, or Y-linked, mutation happening in the Y chromosome. Okay. Under X-linked, it could be a dominant mutation, X-linked dominant, or X-linked recessive. Okay. Y-linked, it has only one chromosome of Y. Okay. So it is always recessive in front of the, you can say, X, but the gene present in Y will not be present in X, so it will be mostly all the time Y-linked dominant. Okay. Y-linked mean mostly will be all the time will be Y-linked dominant only. Under autosome again, if you have, suppose in chromosome number 1 pair, this one comes from the father, this one comes from the mother. We are talking about a gene. Okay, suppose the insulin gene only, susceptibility towards diabetes. So if this insulin gene is fine, this one is not fine, still you won't get insulin gene. You can say diabetic disease. But if both the alleles are recessive, then insulin hormone, the protein is extremely dysfunctional, you may get diabetes. So it could be either autosomal recessive, like in this case, or autosomal dominant. Autosomal dominant. So these are the classifications of, you can say, Mendelian disorders. Okay, autosomal recessive, you have to first nail the disease, whether the disease is autosomal recessive or autosomal dominant, X-linked recessive, X-linked dominant, or Y-linked. We don't call it dominant because it is always dominant, so just we call it Y-linked. Okay, and one more is the mitochondrial inheritance. Okay, that is not talked about much because that is a higher level thing. So because mitochondrial genome is completely different, it's a circular DNA acquired from the mother only, not from the father. So mitochondrial inheritance is also there, then pedigree analysis is there, but those things we won't be talking about here. Okay, so this is autosomal and allosomal slash X-linked diseases and Y-linked diseases. So this is the entire setup. Okay, so now coming on to the mechanism. Okay, let's talk about, you can say, hemophilia. Okay, you might have heard of hemophilia before. So it happened to the British Queen. Okay, so clotting factor 8 deficiency. Okay, so coming on to hemophilia. So hemophilia, if we talk about, it is an X-linked recessive disorder. Okay, X-linked recessive disorder. Now, how did I understand that it's an X-linked recessive disorder? So, number one thing is, if you have two X chromosomes, okay, in a woman, so hemophilia does what? The hemophilia disease will not let the clotting factors be produced. Okay, clotting factors are the proteins which are produced by the liver. Now, when we talk about hemophilia, simple hemophilia, okay, what we say is we are talking about a specific type of hemophilia known as hemophilia A. Hemophilia, simple hemophilia, if it is written in the exam, means they are talking about hemophilia type A. Hemophilia A. There are more than one type of hemophilia. So if we talk about hemophilia A, means we are talking about factor 8, clotting factor 8, which is the protein synthesized by the liver. Now, On this is an X chromosome, okay, that is the homolog of X chromosome, okay, coming from the father. This is coming from the mother. So the thing is, the clotting factor 8 gene has got some, you can say, mutations that make it completely dysfunctional. But if this homolog has proper 8 gene, clotting factor 8 gene which is proper, now still the girl will be able to make clotting factor 8 or not, because one of the gene is working properly, so she will not suffer from hemophilia. So I can call it, you can say, the small x or xh, okay. So still she will be able to code for the coding factor 8 protein, because one of the alleles is dysfunctional, the other allele is still functioning well. Alternate form the same gene present on the homologous chromosome. So other allele is still functioning well. So she is able to code for the protein coding factor 8. But now let's check the another scenario. This is chromosome X from father, chromosome X from mother. Clotting factor 8, you can have some deleterious mutation. Means dysfunctional protein. Same problem here, same mutation here. Again dysfunctional protein. Now, cumulatively, they can never produce, neither this allele is appropriate, neither this allele is genetically appropriate. So, when the transcription will occur, mRNA will be produced. So, mRNA, when it goes and binds to the ribosome present in the ER, rough ER, now what will happen is the clotting factor 8 protein that will be synthesized, okay, CF8, will be dysfunctional from both the alleles. Okay, both alleles are contributing towards production of dysfunctional clotting factor 8 protein. Thereby, the person will have blood clot deficiency problem. Means if a cut or wound happens, a wound occurs, the bleeding will not stop, the bleeding may not cease. Okay, so that is known as hemophilia. So it is X-linked recessive. When both the disease have deleterious mutation, I told you what is recessive if you remember in the first class itself. That recessive doesn't mean you are a weaker candidate, it means you have some mutations which cannot produce the protein. Okay, so that means it is an X-linked recessive disease. If either one of the genes are normal, means dominant, then the one recessive allele cannot mask the effect of the other allele, because the other allele is producing the appropriate or the correctly, you can say, conformationally stable protein. That is what is known as X-linked recessive. Now, if we talk about the genetics of this one, suppose the women can be mostly the carriers. Okay, the carriers means, if I talk about XC, carrier X, means she has one allele which is a mutated allele, one allele is perfect. Suppose she marries a person XY. Here X means he is not hemophilic, she is a carrier. So what could be the probability? XCX, XX, then XCY, then XY. Now in this case, this is a male. Okay, the Y chromosome does not have gene for clotting factor 8. It is only dependent on the X chromosome, and the X chromosome has, it is the XC type, means defective clotting factor 8. What does it mean? The male will be phenotypically hemophilic, right? The male will always be hemophilic in nature or by phenotype, because Y chromosome does not contain any allele for clotting factor 8. So it will not have any competition. So it depends on only X chromosome. If the defective X chromosome comes, so if a male, normal male, marries a carrier male, or sorry, carrier female for hemophilia, so there is a definite 25% probability that if he has a child, okay, that will become hemophilic, and especially if he has a boy, then a male, then 50% of the boys, these two boys out of one is 50. So 50% of the boy or the males will be hemophilic. Okay, and if you think from the male's perspective, you have a hemophilic male and marries a carrier female. So XCY marrying a carrier female, XC, then XCY, okay, XC, then XY. So then you can have female. Only when the hemophilic male marries a carrier female, then only you will be developing or giving rise to a female which can be hemophilic. Otherwise, they cannot be hemophilic. And the males will not be live up to the age of 20 plus because they will die because of multiple internal and external, not only external, internal bleeding may also occur. So it is not a probable event that a male will live through his adolescence and become 20 plus in order to marry a woman, right? And then only women are completely hemophilic. So the probability of women being hemophilic is very, very less. Okay. One more thing is that is why you can say the affected males, okay, the affected males do not transmit mostly to their, you can say, causative, you can say, disease to their male counterparts also, because they will not be able to live only. Okay, so women male, they are not able to transmit. Okay, so once we talk about, you can say, one more thing I'll tell you, that's a very interesting stuff. So if you see the this one only, hemophilic probability, everything. So if you see here, I'll tell you one thing, that males are more often affected than females, because males are needing X chromosome and they do not have any other X chromosome. So whatever X comes, that will be affected. Female have two X chromosomes. So even if this is XC, this one is proper, they will not be affected. Okay. Next thing, the, you can say, affected fathers. Okay. What happens is, they mostly, what they do is, affected fathers, they do not pass to their sons. Mostly they do not pass to their sons. Sometimes they can pass. Okay, because this is the affected father. If it is he is marrying like a child, you can say woman like this, then he can pass. But if the woman is you can say normal, then he won't be passing. Okay, if he is normal. So these are the two characteristics that you should be just thinking about when you think from the conceptual standpoint. Okay, that is the concept of X-linked recessivity. Okay, so is it clear, Ishika, X-linked recessive disease from the conceptual point of view, that why are males more susceptible to the disease because they have only one X chromosome, and why they don't have the genes which is present in X, so they will be vulnerable to that disease. Okay, good. So next up is, we'll talk about, you can say, X-linked dominant. It's a very, very rare disease. Okay, X-linked dominant means two chromosomes are there. This chromosome is appropriate. Okay, appropriate. The molecular basis is different. This is inappropriate. Okay, means mutated. But still, what it has is, it will cause the disease. It will still cause the disease, but you will think that this is a recessive disease. How can I call it dominant? How is it able to cause? Because the mutation is a gain of function mutation. Very, very important. Gain of function mutation. means this protein is not an utter waste. This protein may overhaul the pathway, may overexpress, and once you overexpress something, again you can cause disease. Means the protein which is produced, the mutation is such that suppose this is a receptor. Okay. Now when this protein is produced, okay, it may go and bind to this protein and cause the, you can say, protein mutated may bind to the protein normal, which is this one, and cause the inactivation of this protein. One hypothesis is this one: protein normal becomes inactive, means it has gained this function, gain of function. Another is, suppose it's a ligand for any receptor, so if the protein mutated bound to its receptor, okay, now it will never dissociate. Once it never dissociates, the signaling will keep on going on, and keep on going on incessantly. And then what will happen is you will have too much signaling event that will lead to, you can say, cell death or multiple cell division, cancer progression, anything it can happen. Okay, so this is known as gain of function mutation. In recessivity, what happens is you have loss of function mutation, means the mutation causes loss of function and the protein becomes useless. So the other allele, if it is appropriate, that will take care. But here other allele, if it is appropriate also, it cannot take care, because the other allele which has got a mutation, it's a gain of function mutation. It has overactivated it, like you have produced a Godzilla or a King Kong, right? It is like that. Or you might have watched X-Men, it is like that. The mutation is making you more powerful, and then you can even suppress the normal person. Okay, it doesn't make you any weaker or lesser. It's a gain of function mutation. So that is why this is known as dominant mutations. Okay, the dominant mutations will always express themselves even if the other allele are is fine also. They don't need both the alleles to be dominant. In case of recessive, both the alleles should be dysfunctional. Then only what will happen is, in the homozygous recessive state, only in the homozygous recessive state will it be able to cause an effect. But in case of dominant one, heterozygous will do. Dominant one, heterozygous is enough. Okay, you don't need both to be dominant, because even if one of the allele is faulty, another one is completely fine. The faulty allele will code for a very aggressive protein which has a gain of function mutation in that specific allele, in that specific gene. That is the molecular reason behind dominance. Okay, so understand why dominance can be is always heterozygous, because it's a gain of function mutation. Okay, so understood why is dominance always heterozygous. These things will not be told to you, you can say, by the book or something. They will just say it is dominant, it is recessive, but this is the real molecular reason behind why is it dominant. Okay, it is like learning genetics from the root. Everything, there is a specific reason behind in biology, nothing to mug up at all. So, any doubts in why is it dominant and how does dominant operate in heterozygous condition only? Any doubts here? Okay, so now we'll come to types of mutations. Very important. One is known as types of mutations, means we are talking about point mutations here. Okay, so only talking about point mutations. Point mutations means one base, one nucleotide change only, which I had shown you. So it could be missense, it could be nonsense, it could be silent mutation or null mutation. Okay, these three could be there. So suppose, in order to understand this, let me tell you something about the genetic code. Otherwise, it is, you cannot understand this. Genetic code is in a different chapter, but you need to know the basics of genetic code at least in order to understand types of mutations that can happen. Okay, so genetic code, let's come to it. So there was an Indian scientist known as Hargobind Khurana. He went to US. He was awarded the Nobel Prize along with one more scientist for discovering the genetic code. Now, what is genetic code is? Genetic code is something like, suppose we have this gene, okay? So A, T, G, C, T. So just segregate the entire nucleotides or bases present in DNA into group of three. Into group of three. And these three codons, they are known as codons, means when the mRNA is produced, okay, the mRNA will have U instead of, suppose this is the mRNA. Let's talk about the mRNA. 5 to 3. So mRNA has AUG, CTU, okay, UAG. Don't put UAG here. Let's put CUG, CCU, like that. Okay. So let's. So why am I segregating the code present in the mRNA, which is just complementary of the DNA, in 333 in a group of three? Because and each group of three means each codon. These are known as codons. This triple nucleotide group is known as a codon, and each codon codes for one amino acid. Means it is responsible for addition of one amino acid via tRNA. That process is will be explained to you later on when the chapter comes. So know that this fact that each codon is responsible for a specific amino acid. Okay. Now, why only three? Understand this fact that we need 20 essential amino acids, humans. Okay, amino acids like glycine, lysine, tryptophan, tyrosine, phenylalanine, histidine, methionine, cysteine, isoleucine, valine, aspartate, glutamate, okay, proline, then asparagine, okay, then glutamine. So many are there. So 20 essential amino acids are there. Now, how many bases are there? Four. ATGC or AUGC in case of mRNA. So four bases. If only one base could code for one amino acid, then 4 to the power 1 would have been 4. Means only 4 amino acids would have been coded, which is not enough. You need 20. If the group was of two bases, then 4 to the power 2, 16. Still you are 4 amino acids short. Now the group is of 3 bases. Then 4 to the power 3 is 64. Now you have completed 20 amino acids, but you have more codons. So what will happen is the excess, okay, 20, 64 minus 20. Okay, what remains is the excess number of codons will become redundant, meaning that more than one codon, suppose this one is coding for glycine, so this one is also coding for glycine. So more than one codon can code for one amino acid. Okay. So when more than one codon can code for one amino acid, when more than one codon codes for one amino acid, it is known as degeneracy of the genetic code. Degeneracy. So genetic code is degenerate. One of the characteristics of genetic code or hallmarks of genetic code is it is degenerate. Means more than one codon can code for one amino acid. But at the same time, genetic code is unambiguous. It shows unambiguity, meaning that one codon codes for only one amino acid. This codon cannot code for any other amino acid. Suppose you think that CUG will also code for lysine. No, it will only code for glycine. So more than one codon can code for one amino acid, but one codon A single codon, a single triplet codon cannot code for more than one amino acid. Understand this sentence clearly. That is unambiguity. So CUG cannot code for two amino acids, but CTU, CUG, CCU, these three can code for glycine individually. But individually, one triplet codon cannot code for more than one amino acid. If it is coding for glycine, it will code for glycine only, not lysine or alanine or anything else. Okay, so this is your the minimal amount of genetic code that you need to know. One more thing I'll tell you, genetic code is universal. Means AUG codes for methionine in case of humans. It codes for methionine in case of bacteria and viruses also. In non-chordates also, annelids, arthropoda, anywhere. Okay, that is genetic code is universal. The codon that codes for one amino acid in any organism will code for the same amino acid in any other organism present in this earth. Okay, genetic code is degenerate, it is unambiguous and universal. So these three characteristics you should know before we talk about any kind of point mutations. Okay, so now tell me, Shika, is there any doubt in any of these three genetic code prospects or characteristics? Any doubt here? Why three nucleotides are there? Why a group of three, not group of four, not group of two, not group of one is needed? Okay, degeneracy, unambiguity, and universality. So it must be universal. So is everything clear regarding the genetic code? Okay, good. So now coming to point mutation. Okay, now missense mutation means suppose the CTU is coding for glycine, just like I told you. Suppose you have a mutation here. You make CTU CGU, and CGU is coding for lysine. So what you did, due to one point mutation, earlier there was T was replaced by G. Now instead of coding for glycine, you are coding for lysine. Suppose this is amino acid number 1, number 2, number 3, number 4. On the second amino acid, what happened was the glycine has been replaced by lysine or not. And once it has been replaced by lysine, what happens is the conformational stability of that specific polypeptide chain or protein can go, or it can become dysfunctional, can become, you can say, unstable. Okay, and then the protein itself can become dysfunctional. So this is known as missense mutation. You are changing the amino acid by playing with one of the amino acid in that triplet, or one of the bases in that triplet codon. That is the meaning of, you can say, missense mutation. Okay, now coming on to, I'll draw it again. So when U was changed to T, suppose, this is all assumption. This is changed to, there is no T here in RNA. Suppose CAG. So this one was coding for glycine. Now this one is coding for lysine. So change in the amino acid because of the change in the nucleotide. So this is a missense mutation. Now suppose let's talk about another type of mutation. Again, I'll take the mRNA. Now suppose a mutation occurred. The mutation has occurred, from U you have become C. But again, let's suppose that CUG codes for glycine And you were very, very fortunate that CGG is a degenerate codon which also codes for glycine. Maybe glycine is coded by these two codons, and you are so fortunate that this codon, the mutation occurred, but the amino acid is the same. Even if mutation occurred, will you have any kind of changes in the protein or in the chronological amino acid placement in the protein? No. At the second position here, 1, 2, 3, 4, 1, 2, 3, 4. And the second position here there is glycine. Second position here there is glycine. Even if mutation has happened, phenotypically the protein is the same. Sequentially, protein sequence wise, the protein is the same. Therefore, this is a null mutation or silent mutation, means it has no effect. There is a mutation, but you have been so fortunate that it is uh, degenerate codon mutation, and you have the effect is completely nil. No phenotypic effect. Even if you have a genotypic mutation, but there is no phenotypic effect on your body. That is known as missense, sorry, you can say silent mutation. Another is nonsense mutation. In order to understand nonsense mutation, know for a fact that there are three stop codons. Okay? That is nonsense, means it will completely take the sense out, means it will cause immediate termination of the protein. Nonsense means out of 64 codons, 61 codons code for proteins, 3 codons are UAA, UGA, UAG. These are the stop codons. Means if the RNA polymerase, okay, not RNA polymerase here, if the ribosome is able to detect these, because this is mRNA, now they'll bind to the ribosome and ribosome will walk over each triplet, they'll jump triple. Okay, what happens is during the translocation, there is a factor known as elongation factor G. It is accompanied by GTP. GTP gets hydrolyzed to GDP and PI, and then the ribosome makes a triplet jump from codon to codon. And when ribosome encounters a stop codon, suppose here UAA, okay, it will dissociate from the mRNA and the polypeptide will come out. That is known as termination of protein synthesis or translation process. Protein synthesis is known as translation. Okay, mRNA formation is transcription. Okay, DNA duplication is replication. So these three are the stop codons. Now, suppose the AUG, okay, it has CUG. Okay, so let's make, suppose it was UAC, CCU, CGA. Suppose it's a new strand, okay, and there has been a mutation. Okay, so mutation is again a point mutation, one nucleotide change only. Now UA has become A, from C it has become A, CCU, CGA. Okay. Now once it has become UA, the ribosome will dissociate from the mRNA here only, and the next codons will not be read. So protein is only 2 amino acid long. So it is a complete waste protein. It's not even a dysfunctional protein. It is such a short protein that it is an utter waste. It will cause immediate translation. You can say translation termination, or immediate termination of, you can say, the protein synthesis process, and the polypeptide will be extremely truncated, maybe 1 or 2 or 3. Wherever it encounters the stop codon, it will terminate, and the post codons will not be read by the ribosome at all. Okay, they are an utter waste. These are the utter wastes. You can say from here only it will be terminated. So that is known as your nonsense mutation. Nonsense mutation event. Okay. So always remember this fact, that what is nonsense, what is missense, and what is, you can say, the silent mutation. So now tell me, Shika, is missense plus silent or null and nonsense mutation? These three are clear. These three types of mutations, they are very important for any entrance tests. Are they clear to you? Conceptually speaking, from the genetic code perspective? Okay, good. So these are the proteins that are extremely essential for the, or these are the mutations that will be extremely important and significant in your preparation for any kind of, you can say, logical conceptual examination. So one of the examples I'll give you of missense mutation. Okay. So if you have heard of something known as sickle cell anemia, okay. So sickle cell anemia is a specific type of anemia whereas, or in which the RBC shape, it changes from being a biconcave to a sickle shape. The RBC is a biconcave enucleated lives for 120 days, the erythrocyte, it becomes like this, sickled. So it cannot, you can say, it doesn't have proper hemoglobin molecule, thereby its shape is sickled, and because of this, it cannot go through the capillaries also, because of biconcave, it can pass through, squeeze through the capillaries. Because capillary have 5 to 7 micrometer of diameter, RBC's diameter is almost 8 micrometer. So conceptually, it should not go through the capillary, but because of its shape, it can squeeze through. But this shape will hinder the squeezing effect also, and hemoglobin is also not proper. Okay. So the thing is, this sickle cell anemia, okay, is a very known disease. It's a very renowned disease. Okay. What happens is the hemoglobin molecule in adults, like we have talked about many times, is made up of two chains, two polypeptide chains, alpha to beta 2. This is adult hemoglobin. Alpha to gamma 2, this is fetal hemoglobin. Okay, different polypeptide chain. Then beta 2, epsilon 2, this is Gover 1, embryo. Okay. Then alpha 2, epsilon 2, this is known as Gover 2, again in the embryo. Okay. So we will talk regarding the alpha to beta 2, that is the adult form of hemoglobin. Now, what happens is, in the beta chain, okay, the gene which codes for the beta chain, okay, what it has is, it has a specific mutation. Okay, so the DNA, the mutation is like this. So I'm just explaining to the partial sequence. Okay, so let's suppose this is the DNA. Okay, so CCT, okay, that is the 3 prime to 5 prime DNA I'm talking about. And then GAG, then GAG. Now, what happens is, when you make mRNA 5 prime to 3 prime antiparallel, it will be CCU. So let's make it DNA only 3 to 5, then we go for mRNA. So CCT, then GAG, then GAG. This is the 5 to 3 prime DNA. 3 to 5 prime DNA. Let's first make the double stranded DNA for the beta chain. It will be more clear for you. DNA for the beta chain. So this will be the just opposite, G, G, A, then C, T, C, then C, C. Now mRNA will use this DNA as the template, 3 to 5 prime, and will look like this DNA, except T will be replaced by U. So mRNA will be coded by RNA polymerase. This is the mRNA. This will be your CCU. Okay, then GAG, then GAG. So CCU codes for proline. This codes for glutamic acid. This one codes for glutamic acid. This is the sixth position. Now what happens is there is a mutation in the, you can say, A region. This becomes T. When this becomes T, what will this become? This will become A, right? This will become A. So what we'll have is, when this has become A, so this will become, instead of, you can say, this is T, this will be same as that, so this A will become U. Okay, this will become U, because this will be used as a template. So now if this has A, okay, now make mRNA, so this will become CCU. Okay, from GGA CCU, CAC will be GA's, you can say, complement will what, will be U, so this will become here U, right? Now GUG is not GTG. Oh, sorry, it's not GAG. So GUG is a missense mutation here. So GUG will code for valine. So at the sixth position of the beta chain, you will have valine instead of glutamate. Okay. So glutamate is nothing but glutamic acid. So in biochemistry, ic acid is known as 8. So glutamic acid is glue, okay, or glutamate, like pyruvic acid, pyruvate, lactic acid, lactate, okay. So this is an acidic amino acid, and valine is a neutral amino acid. So because of such drastic changes in structure and in function, the entire beta chain is dysfunctional, and therefore the hemoglobin contained in the RBC will be dysfunctional. Each RBC as such contains almost 278 million hemoglobin molecules, and each hemoglobin molecule can bind to 4 O2 molecules. But here, since the hemoglobins are dysfunctional, okay, they are written as HbS, sickled HbS, and normal hemoglobin is written as HbA, hemoglobin adult. Okay, so it is different from hemoglobin adult. So the RBCs, they attain a sickle, or they attain sickle cell. So this is again an autosomal recessive disorder. If both the alleles for beta gene have this problem, then you will be having this problem. Otherwise, you may not have phenotypic problem, because one allele is fine, it will code for proper hemoglobin. Okay, so it's an autosomal recessive disorder. So tell me, Shika, is there any doubt in this lecture, today's lecture? Any doubt? Any queries? I hope you are able to comprehend everything. And you can send me any sheets if you have, or if your school provides you, or if you encounter any problem. Okay, so we'll solve it out and give you the solutions. Okay, okay, good. So now you can go to school, stay safe, have a nice day. Okay, Sika, thanks a lot. Let's meet in the next class.